In this video, we will be covering these concepts of our immune system, its defense layers. Innate immunity, acquired immunity, in which we will see more details about active and passive immunity, antigens, and haptens. More topics will be covered ahead in the series.
We can start to organize the magnificent chaos of our biology into an elegant, structured grid. The systemic defense layers stack perfectly together. You have physical barriers on the outside, innate immunity in the middle, and adaptive immunity deep within. This three-dimensional matrix tracks cellular components, like T cells orchestrating direct killing and macrophages engulfing threats. Notice how these systemic layers ultimately narrow down into very precise molecular triggers, flowing logically from the initial recognition of a pathogen all the way through to the ultimate resolution. Now, this entire architecture operates across two fundamental paradigms. First, we have innate immunity. Think of this as your inborn resistance. It's present from the exact moment of your birth and is dictated entirely by your genetic and constitutional makeup. It serves as your absolute first line of host defense, acting immediately, often within minutes of a threat. Second, we have acquired immunity. This is a highly specialized, precisely targeted resistance against foreign substances that you dynamically develop over your entire lifetime. Because of its incredible precision, this acquired response takes a significant amount of time, often days to weeks, to fully develop, and it splits further into active and passive branches, which we'll explore in detail shortly. This sequence brilliantly illustrates the immediate cellular alarm of the innate system we just identified. It progresses in 4 calm, highly calculated steps. Step 1 is the breach. Microbe-associated molecular patterns, or MAMPs, which are essentially the physical textures of a bacterium, enter the environment and act as a foreign key. In step 2, the host's lock attaches. Pattern recognition receptors physically bind to those invading MAMPs. Step 3 triggers the alarm. This physical binding sends internal signals that activate transcription factors deep within the host cell's nucleus. And finally, step 4 is the response. The cell immediately produces vital cytokines and enzymes specifically engineered to destroy that invading microbe. Surrounding that alarm sequence are 7 distinct mediators, which form concentric layers of defense. The outermost ring houses our anatomical and physiological barriers, things like our skin, our body temperature, and the low pH acidity of our stomachs. Moving inward, we encounter these cellular defenders, phagocytes and natural killer cells that physically engulf or destroy infected cells. Finally, in the core ring, chemical mediators like cytokines initiate inflammation, and acute phase reactants rapidly increase to further protect the host environment. As we transition deeper into the blueprint, we reach Zoom Level 1, acquired defense dynamics. Leaving the general barriers of the innate system behind, we gently magnify our view to examine the incredibly sophisticated nature of acquired defense.
Unlike the innate defenses you're born with, acquired immunity develops dynamically over your lifetime, and it is characterized by 5 distinct pillars. First, it requires prior exposure. The system remains completely inactive until physical contact with a specific microbe actually occurs. Second is the time lag. Because the body must custom build its weapons, it takes days to weeks to activate. Third is specificity. The resulting defense is highly targeted against unique microbial antigens. Fourth is diversity. Despite this extreme specificity, the system as a whole is active against a vast, structurally diverse range of antigens. And fifth, perhaps most remarkably, is immunological memory. A proportion of the activated T and B cells permanently transition into memory cells to guarantee a highly effective response the next time that specific threat appears.
Comparing these two halves of our biology reveals some crucial trade-offs. On one side, innate immunity is genetic and immediate. It acts within minutes through general pattern recognition, but it possesses absolutely no memory. In stark contrast, acquired immunity is dynamically developed. It requires a crucial first contact and days of time to activate, But the trade-off is that it achieves highly specific targeting and dedicates T and B cells for lifelong memory. This acquired immunity is further categorized by how the host actually participates in the process. Think of active immunity as an internal biological manufacturing plant. The host's own immune system actively participates in building the defense mechanisms directly in response to a stimulus. Passive immunity, on the other hand, is more like a biological airdrop. Resistance is transferred to the host in a ready-made form, requiring absolutely zero active participation or manufacturing from the host's own immune system. What is truly remarkable about active immunity is its sheer durability. Whether it's triggered naturally by a live clinical infection or artificially by a vaccination, the system properties are identical. It does require a significant lag period to build those specialized defenses. However, this wait pays off with incredible duration, providing long-lasting, frequently lifelong protection. This happens because it successfully establishes immunological memory, building a robust bank of T and B cells to ensure rapid responses during any future encounters. Here, we can clearly see how that memory reshapes our survival. Upon first exposure, the primary response is a slow, gradual climb, primarily dominant in IgM antibodies. However, upon a subsequent exposure to that same threat, the presence of those memory cells completely eliminates the time lag. The secondary response transforms into an immediate, overwhelming defense that heavily dominates the threat, and this time, it's heavily reliant on highly effective IgG antibodies. Conversely, passive immunity operates on an entirely different set of rules. It can be acquired naturally, such as the vital transfer of maternal IgG antibodies from a mother to her fetus, or artificially, through the medical transfer of prepared immunoglobulins, like the rabies antibody. Its greatest operational property is speed. It grants the host immediate protection, functioning with absolutely zero lag period. However, this comes with strict system limitations. Because the host's own cells never participated in making it, this protection lasts for only a very short time. To successfully activate this specialized, acquired machinery we've been discussing, highly specific, microscopic keys must be physically introduced into the host. These keys aren't abstract concepts. They exist as distinct, complex, three-dimensional molecular signatures resting physically right on the surfaces of foreign substances. The absolute non-negotiable prerequisite for this activation is immunogenicity. This is the fundamental initial ability of a substance to actively induce an immune response within the host organism. Equally vital, but distinctly different, is antigenicity. This is the subsequent ability of that exact substance to physically react and chemically lock with the highly specific end products produced by that very immune response, most notably the antibodies themselves. As we can clearly see, a true, complete antigen must. Now, the biological potency of a true immunogen is dictated by 5 precise properties. First is size. Larger molecules are exponentially more potent, with a strict threshold generally exceeding 10,000 Daltons, which is a measure of atomic mass. Next is chemical nature. Complex proteins serve as the strongest triggers. Third is structural complexity. Fourth is phylogenetic foreignness. Simply put, the greater the evolutionary distance from the host, the stronger the biological alarm. The absolute smallest unit of antigenicity is the epitope, or antigenic determinant. Notice the sheer difference in scale here. While an antigen is a massive structure, an epitope is an incredibly precise surface area consisting of just 4 to 5 amino acids or sugar residues. Its singular function is to serve as the exact anatomical location that physically locks into an antibody or a T-cell receptor. This highly localized architecture is what determines which cellular defenders can recognize it. Sequential, or linear, epitopes are formed by a single unbroken sequence of amino acids, and these are generally recognized by our T cells. Conformational epitopes, on the other hand, are formed purely by complex geometry during the three-dimensional folding of the protein, and these are generally recognized by our B cells. This precise microscopic geometry introduces a fascinating biological loophole known as the hapten paradox. Haptens are very low molecular weight structures that possess strict antigenicity, meaning they have the exact correct shape to react with existing antibodies. However, because their mass falls vastly below that critical 10,000 Dalton size limit, they entirely lack immunogenicity. The profound limitation here is that haptens are biologically incapable of inducing an immune response all on their own. An inert hapten is instantly weaponized and activated the exact moment it physically bonds to a massive, heavy carrier protein. By combining their mass, they successfully bypass the 10,000 Dalton threshold together, completely transforming an invisible molecule into a powerful immunogenic trigger. Once mobilized in this way, the complexity of these haptens defines their true capability. Simple univalent haptens possess only a single binding site, meaning they cannot cross-link antibodies. But complex multivalent haptens contain multiple binding sites. This unique property allows them to cross-link with specific antibodies. We can now fully appreciate this flawlessly tiered architecture. In phase 1, the breach, MAMPs instantly trigger innate pattern recognition receptors to sound the rapid response physiological alarm. In phase 2, specialization, large antigens above 10,000 Daltons utilize days of lag time to carefully develop a highly specific acquired response. And finally, in phase 3, the lock and key, newly acquired antibodies explicitly target and bind to conformational epitopes mere amino acids wide, neutralizing the threat entirely.